next little unit, we're going to be looking at the renal system. Those of you who have not had lab yet, we'll go over all the parts of what we see up here. This is just a cover slide as an introduction to the renal system itself. We do have to begin discussing how we're going to be making urine by zooming in on a part of our nephron. And just to put it in some context for the moment, all right, we're zooming in on this part right here of this big thing known as a nephron. We're going to be zooming in right in there. We'll learn the parts of them in lab. But I want you to notice from this, even if you haven't had lab yet, is there's this arteriole coming in known as the afferent arteriole. And then there's this capillary bed, a ball of capillaries, which is why it's called glomerulus. And this ball of capillaries will then exit via the efferent arteriole. So this is a bit different, right? Because you have an arteriole capillary bed back to another arteriole. And these black arrows representing the flow of blood in and then the flow of blood out. And then these little green turquoise arrows are going to be called filtrate. And the filtrate will be the fluid that was filtered out of the blood. So we're going to have filtrates. Yeah, not filtrate. Yeah, good. Yeah. There we go. Now, how many liters of blood do you guys have? 5. All right. So that black arrow represents blood going in. On an average of 24 hours, 1,000 liters of blood will come into your glomeruluses. Holy crap, that's a lot of blood, right? 1,000 liters are going to come in. And I'm going to be filtering that fluid. And of course, not all of 1,000 of it is going to be coming out. These green arrows represent 180 liters of that 1,000. So 1,000 come in, we filter 180 liters out. What does that leave left over?
Minus 175.
Minus 100. 1,000 come in, 1,000 minus 180 is? 1,000 minus 180? 880. 880? 1,000 in, 180 out. Oh my God! Okay. Some math will happen. How about 820?
We did it.
Okay. So 1,000 in, we filter 180. Now, the story of tonight and next Monday, besides basic math skills, is going to be if I'm filtering 180 liters out of my blood. Think about urine formation. You guys do not pee 180 liters. You pee about 1.5 liters a day. And dare I ask, 180 minus 1.5, right? All that extra fluid has to be put back into the bloodstream. And that's the part of tonight and Monday's lecture, to figure out how we go about doing all that. So this is an overall schematic diagram of how we're going to be doing this. It is not anatomically accurate, as the first lab group has been warned and realizes, but it's more like a functional diagram. There's my blood coming in, right? There's my turquoise arrow for filtration. Now, I've got 3 major functions that can happen in my nephron. Glomerular filtration, tubular reabsorption, and tubular secretion. Look at that middle purple one for a minute, tubular reabsorption. It's not tubular absorption, it's reabsorption. Meaning it's the second time that stuff is going back into the bloodstream. It's the second time that stuff is going into the blood. Now they color-coded it for you with the diagrams. So that kind of a lightish green color, right? Where do I have any lightish green arrows? Light green. I got one right there and that's it. Which means that's the only place that glomerular filtration takes place, at the glomerulus. But then my purple and gold arrows happen everywhere. And so we have some terms to define, right? This is known as reabsorption. The purple is reabsorption. We're going from the filtrate back into the blood. From the filtrate back into the blood, filtrate to blood, filtrate to blood, filtrate to blood. The opposite direction is known as secretion. From the blood into the filtrate. 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 The things that are small enough to be filtered will be my little green arrow here. But if it's too big to be filtered, but I want it to be in my urine eventually, I have to use secretion. And larger substances will be secreted into the filtrate. We'll be looking at all of these today and on Monday.
So which is the part that's the small things?
Glomerulus filtration,
Oh, okay.
because that's where you're going to have your fenestrated capillary. All right, let's zoom in on that. If you remember, when we had our lab discussion about what actually takes place in the capillary bed, and I said you guys have to understand this because it's the basis for what's going to happen in the renal system and in digestion. You have to understand the idea of capillary hydrostatic pressure, blood colloidal osmotic, because guess what? We're back at it again. The exact same concepts. It's a capillary bed, and we're doing filtering. Well, what drives the filtration? 3 different forces are working together with this, not just 2. Now we have 3. So there is my hydrostatic pressure of the glomerulus. That's the pressure in the capillaries. That's your blood pressure in the capillaries. I'll even allow the first row to answer this question. Does anybody remember the pressure of the capillary hydrostatic at the arterial side of the capillary bed?
35 millimeters.
35, very good. What's this number? 60. This is a high pressure capillary. Way higher than normal. So I've got 60 units of force trying to push my plasma through those pores of my fenestrated capillary. Now, I also have osmotic pressure in the capillary, right? That's the solutes that are too big to be filtered that are trying to keep the water in the capillaries. So these two are the same as our capillary bed discussion, right? The pressure of the blood trying to push the filtrate out, and the solutes inside the capillary trying to keep the fluid from leaving. There's one additional force that's in here, and that's this little HP, the hydrostatic pressure of the capsule. In other words, it's sort of this idea, if I can demonstrate this. All right, I'm going to make my capsule, right? That's going to be this thing here. It's more like a kidney. And my tube is going to come out this way. Then we're going to draw, I'm just going to represent a big circle as my ball of capillaries. And then there's that little efferent coming out like this. And there's the big afferent kind of coming in to form this. So all of this is my capillary bed. We're pushing fluid out, right? That's my blood pressure. There's proteins inside, right, trying to keep the fluid from leaving. But then this is a hollow space that's full of fluid now. There's filtrate in here. Hey, I spelled it right this time. Now, that fluid is pushing against the outside edge. That is pressure. That fluid is trying to push its way back into the capillary bed. The osmotic pressure is the solutes in the capillary trying to keep the water in the capillary. But this is the force outside trying to push its way back in. So I've got an outward force trying to push in, I have the blood pressure trying to push out, and then the solutes in my capillary, right, are trying to keep the water in. Not pull it out, but actually trying to keep it in. When I look at all 3 forces working together, right, 60 units of force of blood pressure trying to push the stuff out. I've got 2 forces really, in a sense, trying to prevent the fluid from leaving. And 32 and 18 is 50. All right. 60 minus 50 is 10. Right there, right? Very good. Okay. Right? We have 10 units of net difference. And that's the actual filtration pressure. It's 10 millimeters of mercury. Now, what would happen to that 10 if the blood pressure in the glomerulus went from 60 to 70. Don't say anything out loud. What happened to the net filtration pressure if that became a 70 rather than a 60? What does the number become? 20. Perfect. Fantastic. What if that 60 became a 50? Are we at 0 then?
Yeah.
All right. And that's going to be our concern. We are not concerned about increases in blood pressure. We are concerned about drops in blood pressure. And we have lots of mechanisms in place to maintain that 10 millimeter difference. I don't care about increases. I'll just pee more. But I have to make sure I have at least a 10 millimeter unit of force of net pressure. And we'll be looking at a bit of ways of adjusting that tonight. All right. Just to put it in some perspective in terms of how this is going, all right? We're going to see a little more of this in lab tonight, but I want to remind you of the idea of fenestrated capillaries, right, which have large pores. The turquoise arrows are the path of filtration through the wall of the capillary, right, into my empty space right here on the whiteboard. If we zoom in on it, this part right here is the inside of this capillary. Then there's the wall of your capillary, right? And those are the pores of fenestration. The green, whatever, what color is that? Limeish green, whatever, right? That's the path of filtrate. Notice what is not leaving. Large proteins. What is an erythrocyte? Oh, okay, red blood cells. What's a leukocyte? Yeah. So in other words, our formed elements aren't leaving. They're too damn big. Only the liquids and small soluble particles and small proteins and sugars and amino acids can actually be filtered through. So those greenish arrows, right, are due to my 10 units of net filtration pressure. They're due to that. All right. This is me and my wonderful PowerPoint skills. We need to look at ways of changing pressures. We got to get some terminology set up here. So here's my afferent arteriole, right, with that black arrow they showed in your lecture notes, bringing blood in this way. I know it's 1,000 liters are coming in. We're going to be producing overall 180 liters of filtrate. Are going to be coming out. Well, we have a term to describe this filtration process. We have terms for everything. And it's known as the glomerular filtration rate. Now, to help you with this, you got to think about the use of the word rate. It's about something per time. So in other words, it's going to be an amount per time. Now, I can talk about it in 24 hours as 180 liters of glomerular filtration. Or I can talk about it in a more common sense way, a per minute basis. Your glomerular filtration rate is also known as GFR. That's the abbreviation for it. Right? Glomerular filtration rate. In a daily basis, it's 180 liters. So we can look at GFR, right, per day equals 180 liters. Or I can do GFR per minute. That equals 125 milliliters per minute. Does seem like good exam question stuff? Yeah, sure does. Okay, hint, hint. Okay. Now, I want to maintain this. My GFR is due to that 10 units of net filtration pressure. If my blood pressure drops, that 10 can become a 9, right? Or an 8. If those numbers drop to an 8 or a 9, then this is going to drop as well. I don't want that. I want to maintain 125 milliliters per minute. I want a nice normal GFR of 125. This is to show us how we can make adjustments in case I get changes in my blood pressure or my glomerular filtration rate. So what I tried to draw with you guys is a normal structure. There's my afferent, my efferent. Notice what I did. I made the afferent really big, the efferent small in comparison. It's supposed to be that way. Because think about the idea of diameters and flow, and the things I'm trying to drum into you guys. If I had a large diameter thing and a small diameter thing, the large has large flow, low pressure. The more narrow has probably more pressure going through it. This is my glomerulus. Now, the idea is how the afferent and efferent arterioles result in glomerular filtration rates. What did I do to the afferent in this diagram compared to what it used to be? So what happens to the flow of blood into glomerulus? It increases. What happens to pressure in the glomerulus? It increases. GFR increases.
Increases?
It increases. You got more flow in. I didn't change the efferent, so more goes in, right? It's going to build up the pressure. So if my blood pressure were to drop, I could dilate the afferent, cause more blood to go in. That would raise the pressure, and I've solved my problem. Because that will cause an increase in GFR. Now, look at the one on the top right. I didn't change the afferent, but I did dilate the efferent. So the same flow is going in, but now it's easier for it to leave. That causes the pressure in the glomerulus to go down. It's easier for it to leave. And if the pressure goes down, what happens to GFR? If the pressure goes down, what happens to GFR? It's going to go down, exactly. So if I want to lower a elevated GFR, I can simply dilate the efferent, which would lower the pressure in the glomerulus, and that would make the GFR go down. This is all happening subconsciously. Let's look at this one. See if you guys can figure out what's going to happen to GFR. Don't say it out loud. Compare the afferent and efferent to the normal one. Remember diameters and pressures, diameters and flow. What will happen to GFR in this new image? Is GFR going to stay the same? Will GFR increase or GFR decrease? How many say it'll decrease? How many says it'll increase? The increases are right? Because we haven't changed how much goes in, but we made it harder for the things to leave. So we get like a traffic jam inside the glomerulus. How about this one? Compare the afferent and the efferents to normal. Remember, large diameter means more flow. More narrow means less flow, harder to leave. What happens to GFR in this case? Goes up even more. Because now I made the afferent bigger, so I've got more blood coming in, and I restricted its ability to leave. So now, even compared to this one, this causes a greater increase in GFR. So one of the ways to do this is, if I dilate the afferent, the GFR go up? If I dilate the afferent, the GFR go up? Yes, good. Thank you, ladies and gentlemen. If I leave the afferent alone, but I dilate the efferent, what happened to GFR? Down. Went down, right? So keep the efferent normal, dilate the afferent, GFR goes up. Keep the afferent normal, dilate the efferent, GFR went down. Over here, keep the afferent normal, constrict the efferent, GFR went up. But if I dilate the afferent and constrict the efferent, I can really jack it up. I get a big increase in GFR. So we dilated that one, then we dilated that one. We constricted this one. We constricted this one and dilate that one. What's missing? Well, what if I dilate the afferent and left the efferent alone? Will GFR go up or down? It's going to go down, right? Because less blood can go in. I haven't changed its ability to leave. There's just less now going in, so the pressure lowers. Now there's one that's not up there. Which one's not up there?
Constriction of both.
You constrict both, right? Constrict the afferent and the efferent, which will really reduce your GFR, because less goes in, and it'll help the fact that there's the constriction there. Your kidneys will do that when you go running, and we'll talk about that a bit later. The idea is that by alternating the afferent and efferent arterioles, I can alternate the pressure in my glomerulus. Now, if I alter the pressure, I can alter my glomerular filtration rate, which we'll get into a bit later. You want to keep it at 125. And these are things we can do if my number goes up or goes down. I want to bring it back to normal. All right, we're going to talk what happens in the very first tube. So, if we're making a quick little diagram of our glomerulus, I'm just going to do it like this for the moment, a schematic diagram. This is the proximal, bless yous. That's this thing right here. We are talking about that part of my path. My filtrate is now in here. We know the concentration of filtrate is measured in milliosmoles, and the osmotic value is 300 milliosmoles of filtrate. And that's due to all the solutes and volume of filtrate in there. Now remember, reabsorption is the idea of, right, going from the filtrate, right, into the peritubular capillaries, going back into the bloodstream. So of that, I'm going to get about 60 to 70% of the volume gets reabsorbed. So 60 to 70% of that 180 liters gets reabsorbed right at the very beginning. Of all the organics that are in your filtrate, such as glucose, amino acids, and some other little things, 99% of it also gets put right back in the blood. What you want to think about is, here's my filtration, here's the glucose that was in my blood. The glucose got filtered into the filtrate, and we kind of say, hey, no, we need that. We're going to put it right back in the blood. It just came out, we're going to put it right back in. Or you do 99% of our organics. Simply because we can't avoid them leaking through the filtration process. Other solutes like electrolytes are also going to be reabsorbed. We're going to see how we do that in a moment. And really, since water is the majority of your plasma, which is the blood component, and this is really just filtered plasma, then of that 180 amount, about 108 liters of water are going to be reabsorbed in a day right at that proximal convoluted tubule. So the main job of the proximal is reabsorption. That's its main job. It does a little teeny bit of this stuff called secretion. Its main job is to reabsorb. Ms. Tassin.
You answered it.
I answered it, okay. See, I'm a mind reader,
Okay.
okay. All right. Let's look at that process of reabsorption. And we get our bearings. So there's my little glomerulus, right? There's the capsular space. So that capsular space in the PowerPoint slide is this space right here where my word filtrate is. That's that space right along in there. And there's the beginning of our proximal convoluted tubule where my 180 liters symbol is. That's this tube right here. I can see that it's made up of simple cuboidal epithelia. I can see lots of microvilli, which we will also address in lab tonight. There is my peritubular capillary, right? And it's right next door. I mean, it is almost on top of those cells. So there's the blood that did not get filtered, right? There's the plasma that did not get filtered. The little yellow things are the solutes that did not get filtered. Here comes my filtrate moving through the proximal convoluted tubule. If the stuff is moved from the filtrate, right, through the tube, heading toward my capillary, but it's doing so between neighboring cells. That's called paracellular transport. If the stuff that was filtered, that I want to go back into my blood, goes through the actual cells themselves, literally, that's called transcellular transport. And we have to use protein channels to do this. Now, besides the size, this idea right here is we want to really embed in our minds over the weekend. All of your pumps, what do pumps imply? If you hear the word pump, what should pop into your head? ATP use. They're always on the basal side. They're not on the apical side. Meaning, all the pumps are on the bottom part of the cell. Remember, apical is the surface that faces the lumen. My pumps will always be found on the basilar side. So there's my pump, and that's the basilar side of this cell, and this is the basal side of that cell. So if the stuff moves between the cells, it's called paracellular. If it moves through the cell via some kind of a transport mechanism or a carrier molecule, then it's called transcellular. The purple line shows you reabsorption, right? And the goldish yellow is showing you secretion.
So obviously, John, in the diagram, it shows channels of some kind on the apical side. So
There's some there, yeah.
yeah, but you're saying they're always on the basal side.
No, I'm saying the pumps are always on the basal side. I didn't
but
say
channels and
Can be on either side.
Channels could be on either side. Okay,
Yep.
thank you.
they could be on either. All right, now let's actually look at some of those channels and pumps. This is a zoomed up portion of the proximal convoluted tubule. We know that because here's my diagram, right? This diagram that you notice is my little goofy diagram on the whiteboard. And that purple region, right, is this proximal convoluted tubule. And they did a cross section for you to see what the tube is made up of, right? Just a circular pattern of cuboidal-like epithelial cells with lots of microvilli. They took one of these little cuboidal cells and put it right here. So there's the apical side, there's the lumen with the filtrate, that's the yellow portion of the slide. This is the basal side of that cell, and there's my peritubular capillary. So it's just this turned upside down, right? That capillary bed is this capillary bed right here. Now let's keep it simple for the moment. Let's ignore everything except for that leaky channel right there. Ignore everything else for the moment. Remember, leaky channels are just a transport protein that don't need a stimulus. They just leak all the time. There's no door to them. Now, nobody's saying anything. But why is sodium going from the filtrate into the cell? And the answer is your trivial pursuit default answer. Everybody, come on, you gotta start chipping in here. It's going from regions of higher to regions of lower. That's why anything does anything, anywhere, anytime. Always higher to lower. It's pressure, ions, concentrations, I don't care which. That's always why, if you remember that, it's easy to figure things out. There's less to memorize. You just know, oh yeah, well duh. It's a concentration gradient. Now, if I simply just let this happen, eventually, you guys would all tell me it's going to reach equilibrium, which is right. So how do I prevent equilibriums? I have a pump on the basal side that continually pumps the sodium out. So I never get a buildup of it in the cell. I always maintain a region of lower compared to the filtrate. And that is your friendly little sodium-potassium ATPase pump. 3 sodiums out, 2 potassiums in, 1 ATP. You should know that better than your last name. Okay? 3 sodiums out, 2 potassiums in, 1 ATP, like a carnival ride. Right? Now, that always allows me to pull sodium out, which always allows sodium to diffuse into the cell. The next rule to help you with renal physiology is a nice little rule. Wherever sodium goes, it has a puppy that wants to follow it. And that puppy is called water. Wherever sodium goes, water wants to follow. Because sodium creates hypertonic environments. Right? And osmosis always wants to dilute the more hypertonic. So when sodium comes in, we're making this more concentrated. And the water wants to follow that and try and dilute the inside of the cell. But then you pump the sodium out, and so the water says, well, fine, I'll follow you into the bloodstream then. That part makes sense? Yeah? Okay, now, just humor me. What is this membrane made up of? What's another name for a plasma cell, or I'm sorry, a plasma membrane, or a cell membrane? What's another name for it? A semi-permeable membrane, right? What's it made up of? Phospholipid bilayers, hydrophilic, hydrophobics. Is water polar or non-polar? Can polar things pass through a cell membrane?
Yeah.
They need help, right? So really, books shouldn't say that water can diffuse through a cell membrane. I mean, it can, all right? Let's make this a phospholipid, another phospholipid. This is a part of my cell membrane. They're not anchored to each other like intercellular connections are. So I could be a water molecule that just gets, that just kind of basically does this. Move! And I can work my way in. I would not want to depend upon that to save my soul. So there are water channels. There are pores in the membrane. They're called aquaporins. And they allow water to pass through them. There are 10 different kinds of aquaporins. So when they're showing you that little blue line crossing the cell membrane, right? You should envision someone like John taking really long fingernails on a blackboard and just creating squeaky symbols and go, oh my God, what they really mean is aquaporins are probably there. And the water is passing through the aquaporins. Same thing here, happy aquaporins. Now, let's try and follow this. As sodium is diffusing from the filtrate into the cell, the water is going to be following it, right, from the filtrate into the cell. If we simply just watch the sodium in and the water following, sodium in, water following, what happens to the volume of my filtrate over time? It's going to go down, right? What happens to the concentration of everything else in that filtrate? It's going to go up, relatively speaking, right? Because we're removing water from that lumen. That increase in concentration of other things drives their facilitated diffusion. Because they become more concentrated, relatively speaking. So all other things that are in there, right, can also then be brought into the cell due to facilitated diffusion, such as this thing right here. That is a carrier. It's called a sodium glucose co-transporter. It does not require any ATP whatsoever. It simply works by John's rule of when chemicals bind to proteins, proteins change their shape. But it's dependent upon the concentration of sodium. It's actually using sodium's desire to go into the cell. That way I can get lots of glucose into the cell. And there's a separate carrier on the basal side. It's a different one than this one. Then I can facilitate the diffusion of glucose out of the cell.
Which one was the sodium glucose transporter?
The one that uses sodium and glucose? That one right there, Dave.
The green thing.
The green thing, yes, exactly. All right, so as sodium leaves, right, water wants to follow. With water and sodium leaving the filtrate, that makes this fluid more concentrated. Everything else, concentration seems to be going up, relatively speaking. That drives their facilitated diffusion. And notice how we actually got two different ways of getting sodium into the cell. A leaky channel and this sodium glucose co-transporter. That really helps to encourage water coming in. And this is why we get 108 liters of water reabsorbed in just the proximal convolute tubule. It's also why we get 99% of all of our organics to be reabsorbed. Mainly by water leaving, everybody else becomes more concentrated, and that drives their facilitated diffusion. We are not going to worry about, for the moment, the sodium hydrogen exchanger, or hydrogens moving, or this bicarbonate. We're not worried about any of that right there. We're only looking at sodium in, water in, and glucose in. Ignore everything else. All right, I already have enough deer in the headlights to look, so we'll give you guys a 10-minute break. All right, get some cool air.
And
Take a look at this at the last minute of the break and make sure you kind of get the basic idea. If not, we can try and patch that understanding before we go on to the next structures.